Okay, everyone, welcome to Chapter 6 in your Psychology 109 Biopsychology class. We are going to talk about the visual system in Chapter 6. So, one of our sensory systems that is probably the most remarkable of all of the sensory systems. We still have a lot to discover when it comes to the visual system. Even the way that the eyes work, the retina, the pathway to the primary visual cortex. We're going to talk a lot about it, but again, there's lots of detail that we're not going to go into discussing this chapter, and there's actually going to be a couple chapters or a couple pages that I actually leave out of the book as well, because it's just more in an advanced biopsychology class and not so much the intro level. So let's go ahead and get started here. It says, what do we see? Okay, here's the interesting part about it is that it's not like a camera. Your eyes are not cameras. Your brain is not like a memory card that you would put in the camera. Okay, so the eyes pull in these images, but the images are then torn apart into little pieces, because we know that the primary visual cortex is specific for different types of objects, like vertical lines, horizontal lines, and so on. So it doesn't see just a bicycle. It doesn't see just a picture of a house. And there's good reason for that, because then when we were to look at another house that was dissimilar, you would, as you grow as a child, you're like, well, no, that's not a house, because that wasn't the house that I saw in this picture. And you'd have to learn all of these different houses. But that's not the way our visual system works. A visual system works upon, vertical, horizontal lines put into certain arrangements, so that your brain says, well, you know what? I think that's a house because of this and this and this. Okay, so it says here an upside down two-dimensional image is transformed into a three-dimensional world that we perceive. Okay, so first of all, we have to talk about some of the basic anatomy of the eyeball itself before we go to the cortical levels. All right, some specifics about the energy. Okay, the light energy itself. It says here, no species can see in the dark. That's a misconception that animals can see in complete darkness, and that's not true. Without light, there is no sight. Okay, light itself is particles of energy, photons, and also waves of electromagnetic radiation. Specifically, it's the wavelengths between 380 and 760 nanometers. Okay, when we speak of wavelength, we speak of color. So wavelength, think color, and intensity of light is where you think bright. Okay, so here's the spectrum, figure 6.2. Okay. And you can see that the wavelength for visible light, and that would be for a human. Other animals are different. They can see things that humans cannot, but we are mostly between that 400 and 700 nanometers that you see there. Okay, and you see the colors represented by the different wavelengths, and that's why you think wavelength and color together, whereas intensity is brightness. Okay, so this energy coming in from our environment, these wavelengths of these photons, okay, light energy, comes in through the pupil, and the pupil is an opening in the eye. Okay, the pupil, as you know, changes size. So when you're in bright light outside, your pupils would be small because your system doesn't need as much light to still focus on objects, acuity, okay? But when you go inside and there's not as much available light, your pupils are going to widen, open up to try to invite more and more light inside so it can work appropriately. Okay. Sensitivity is the ability to see when light is dim, and then acuity is to see in detail. So we'll talk about these terms many times when we're talking about, let's see, the fovea. The fovea is specific for acuity, okay? But the peripheral portions of your eyes, where you primarily have rods, are going to be sensitive to light, which means that they're going to try their best to render images to your brain, even though there's very limited light available. You also have the lens. Focuses the light on the retina, and this lens is moved back and forth by ciliary muscles. And the movement of the lens is called accommodation, the process of adjusting the lens to bring images into focus. So let's look at the eyeball itself here. Okay. One of the things that we didn't discuss was the cornea. The cornea is actually the outside covering. It's a protective layer for the eye. Okay. But then you see that you have the pupil where the light comes in, and then it is refracted by the lens, moved back and forth by those eye muscles, the ciliary muscles, and then that image is projected back to the back of the eye to the retina. Okay? And we'll talk more about retina, optic nerve, blind spot, fovea in just a second. But this is why that first slide said, well, it's just a two-dimensional upside-down object. How do we see the rose the way it's shown here in the picture on the far left if its representation is that upside-down rose in the back of the retina? Okay. All right. Let's talk more about the retina. The retina, in a sense, is inside out. What that means is that light passes through all of these different cells in the retina. It's actually transduced or absorbed by the rods and cones, which are in the very, very back part of the eye. And then those electrical chemical signals are sent forward through all those cells, amacrine cells and so on, that we'll talk about in a minute, and then out through the optic nerve. Okay, so there's a vertical pathway, receptors to bipolar cells to the retinal ganglion cells, and there's also a lateral communication that is facilitated by horizontal cells and amacrine cells. So let's look at these in a little bit more detail. Figure 6.5. Okay, so you see on the very left-hand side the light. The light is coming in and actually passes through the retinal ganglion cells, the amacrine cells, bipolar cells, horizontal cells, and then it's picked up by the cones and rods at the very, very back portion of the eye. Okay? The rods and cones, these are the receptors. Okay? These are the neurons, if you want to think about it. Okay? And so we're going to talk a lot more about what they do. But when they're activated by light or no light, then those signals, that electrical chemical signal, is then stemmed from the rod and cone cells and pushed towards that light arrow. Okay, so you can think of it as the neurons, those cones and rods are, you have action potentials. Those are being then sent to those horizontal cells, bipolar cells, then the bipolar cells to the amacrine cells and the retinal ganglion cells, and then the ganglion cells out the optic nerve. Okay, so that's why they're saying it's kind of a backward approach. The only system in the body that works this way as well. Okay, the blind spot. You saw that in the diagram just a minute ago. There are no receptors where information exits the eye. So the blind spot is where the optic nerve, right, where those cells are exiting the eyeball. So there can't be any receptors there. So you would think to yourself, well, whenever we're looking around, there should always be a little black dot. In our visual field, there should be a little black dot, and it moves around as our eyes move around. But that doesn't happen. And it doesn't happen for the because of the process of completion. And so this is why there's this integral play between the sensory organ, in this case the eye, and the brain, and specifically the primary visual cortex. So the primary visual cortex can interpret things that really is not being picked up by the sensory organ, in this case the eye, at all. So we actually interpret what we see in our environment, and it can be much different than what we are actually looking at. Okay. The fovea, this is where the high acuity area lies, because at this fovea you only have cones. We'll talk about that in a little bit. So cones are going to be specific for high acuity. It happens to be that the fovea is right in the center of the retina. So guess what? If you want to really focus on something intently and read something, study something, you're going to put it right in the middle of your visual field, because that's where that information is coming into the eyeball and centers on the fovea. Here's another representation here. The fovea is the indentation at the center of the retina and is specialized for high acuity vision. Okay. All right, so what are these cones and rods that I've already discussed or talked about a couple different times? It says here cones. When you think cones, you think the photopic vision. Photopic vision. When you think rods, you think scotopic vision. Okay, so cones, high acuity color. I always think cones and color. If I have cones and color right, then I can associate high acuity with it and daytime. Okay, so that's the way I keep them straight. Rods, good for nighttime. They're high sensitivity, allowing for low acuity vision in dim environments. Okay, so the rods are good when it's when you're in a dim environment. They're trying to pick up as much light as possible to make sense of your surroundings, but they don't have the capability of providing you that perfectly clear picture of what you're looking at. That's going to be due to your cones. There's more convergence in the rod system, increasing sensitivity while decreasing acuity. And again, here's another little thing I mentioned a little bit earlier. Only cones are found at the fovea. Okay, so fovea, high acuity. Why? Because they're all cones, and cones are all about high acuity color. Okay. Now, a little bit more on that convergence topic. You see here that up at the top you have a cone. Remember, high acuity color cone. Low convergence, which means that in some cases you have one cone that is sending a message to one bipolar cell and one retinal ganglion cell. Okay. Below that, if you see, it actually depicts 1, 2, 3, 4, 5, 6, 7, 8. So you have 8 rods influencing 4 bipolar cells to then converge on one retinal ganglion cell. So with the rods you have high convergence. With a cone you have low convergence. So again, going back to the low convergence and cone, that retinal ganglion cell can be very, very specific to what you're seeing. Okay. At the bottom, you have several rods that are influencing one ganglion cell. So it doesn't have that specificity. Okay. Now it works. You want the high convergence for rods because they're trying to pick up as much information in that dark, environment. Okay. So it's beneficial for it, but it's not influencing just one of these ganglion cells. So it's not going to be very, very specific. Okay. Says when a retinal ganglion cell that receives input from hundreds of rods changes its firing, the brain has no way of knowing which portion of the rods contributed to the change. Although a more intense light is required to change the firing of a retinal ganglion cell that receives signals from cones, when such a retinal ganglion cell does react, there is less ambiguity about the location of the stimulus that triggered the reaction. Okay. I hope that helped a little bit. It's kind of a hard concept. Okay. The distribution of rods and cones, you can see here. Look at that. Fovea, right in the middle. There is no blue in the middle. Okay. There is only that red, and those are only cones. Okay. But most of the rods and cones are within, 20 degrees of the fovea. And then they start going down on the sides. Once you get to the lateral most part of the retina on both eyes, you're going to have primarily rods there, very few cones. Okay. It's actually one of those things where it's kind of interesting. If you were to kind of investigate this yourself, if you use the peripheral of your eye, try to look as far left as you possibly can or as far right as you possibly can. In a dim environment, you would have a little bit more detail because you'd have more rods in that area. Okay? Not that we're going to go around in the dark and then be trying to stare out our the most lateral portions of our eyes, but physiologically, that's what's true. Okay. We have spectral sensitivity. Lights of the same intensity but different wavelengths may not all look equally bright. A spectral sensitivity curve shows the relationship between wavelength, and there are different sensitivity curves depending on if it's photopic or if it's scotopic. And let's look at this. Okay, so what we're looking at here is it says under scotopic conditions, the visual system is maximally sensitive to wavelengths of about 500 nanometers. You can see that scotopic at 500, that's where it peaks at 100%. Thus, under scotopic conditions, a light of 560 nanometers would have to be much more intense than one at 500 nanometers to be seen as equally bright. So for photopic, you need to be at, let's see, I think 560, 550. But with scotopic, you need to be at 500. That's what the peak is for that maximal sensitivity. Okay. What about eye movement? We continually scan the world with small and quick eye movements. These are called saccades. Saccades. These bits of information are then integrated. If we were to stabilize the retinal image, let's say you anesthetize somebody, of course most of the time they're going to do this with animal species, and they're looking straight ahead, they will soon see like a kind of a dark cloud right in front of them. Okay, so they wouldn't see anything. Remember how I mentioned that the information that comes in, and then you have that blind spot. Okay, well, the reason why you don't see that blind spot, that little dot in your visual field, is because your eyes are always constantly moving and shifting just a little bit back and forth. These small movements. And because it takes some time for the cortical regions of the brain to interpret that, you don't see it. Okay, it's kind of like those those drawings on a piece of paper where you draw the little stick figure and then you flip the pages through and you see this little person moving. Okay, you don't see the individual images. You see the image as moving, as more continuous. Okay. Okay, transduction. We are going to be using this term for the next couple chapters, maybe 3 chapters. Transduction is the conversion of one form of energy to another. Okay, so let's think about this. For the visual system, what we said the physical energy was wavelengths of light. Okay, these photons, light energy. All right, so that is our physical energy in the environment, and it has to be transduced. It has to be changed into an electrical and chemical signal that can be understood by the cortical regions of the brain. And so conversion of light to neural signals are performed by the visual receptors, and those receptors are going to be the rods and cones. Pigments absorb light. Specifically, we're going to talk about rhodopsin, which is a red pigment in cones. An absorption spectrum describes spectral sensitivity. Okay. And here's where we talk about, here's your rhodopsin. The sensitivity of human vision to different wavelengths under scotopic conditions. Again, that 500 nanometers is pretty much the 100% mark. And then in green, you have the ability of rhodopsin to absorb different wavelengths. Okay? And you can see that it is a perfect relationship there. Okay? And the sensitivity, of course, wavelengths under the scotopic, the reason for that is only because of the absorption of rhodopsin. So those lines would have to mirror one another. Okay, so here you go. Rhodopsin, the pigment, is the pigment found in rods. It is a G protein link receptor. Remember back in our chapter 4, it would be neurophysiology. There were 2 different ways of activating neurons. We talked about the protein channels, and then we talked about G proteins. And then we didn't go into a lot of detail on G proteins. I focused mostly on the gated channels, proteins where the sodium is flowing in, etc. But we'll look into this one a little bit. Okay, so in the dark, it says here sodium channels remain partially open, releasing glutamate. When light strikes, the sodium channels close. So the rods are hyperpolarized, inhibiting the glutamate release. All right, so let's look at this. The figure 6.12 is much easier for us to understand. On the left-hand side, in the dark, okay, number 1, rhodopsin molecules are inactive. So in the dark, the rhodopsin molecules are inactive. They're actually red. You see them as a red color. In light, they bleach out. Okay, so rhodopsin on the left, they're red. Sodium channels are kept open. What do we know about that? Well, remember, resting state of the neuron, slightly negative, negative 70 millivolts. Sodium is a positive ion. Concentration gradients, electrostatic gradients, it wants in. Sodium floods in, right? And partially depolarizes them. It's going to cause this rod to become less negative. Remember, if it hits negative 65, what's going to happen? You have that action potential down there at the bottom. It says continuously releases glutamate. And glutamate is one of the top excitatory neurotransmitters. Okay, so we went through that whole process. This is actually our first attempt of utilizing what we learned in our neurophysiology. So now we can apply. Okay? And remember all those times where I was telling you that, if you don't understand, the resting state of the cell and the action potentials, the negative 70 millivolts to negative 65, the difference between hyper and depolarization is a big one. Okay, then you can apply it throughout the rest of the course. Okay, so let's look to the right in the light. So light bleaches rhodopsin. As a result, the sodium channels close. Now sodium can't get in, and as a result, the rods become hyperpolarized. So now they're becoming more negative, which is going to make it less likely for it to fire. So at the bottom, the glutamate release is reduced. Okay, so in this case, rhodopsin, when triggered by light, is an antagonist for glutamate. Okay. You might want to pause and rewind that a little bit and go over that again, but that's a perfect example. Okay. Now, from the retina to the primary visual cortex. There is a large portion of this that I did leave out of the chapter. So I think it's far more important for you to understand the visual pathway without getting into some of the other details with the columnar structures of the primary visual cortex and so on. Okay. The retinal geniculate striate pathway includes about 90% of axons of the retinal ganglion cells. Remember, those are the ganglion cells coming out of that blind spot. The optic nerve goes to the optic chiasm, then the optic tracts back to the primary visual cortex. Okay, the left hemiretina of each eye, or the right visual field, connects to the right lateral geniculate nucleus, and then the right hemiretina, left visual field, connects to the left lateral geniculate nucleus. Guess what? That lateral geniculate nucleus should come as no surprise to you. Okay? Remember, it is a part of the thalamus, and we said that the thalamus was a switching station. So guess what? This is physical energy coming from your environment through the sensory organ, which is the eye, and it hits that thalamus, and the thalamus has to tell the brain what to do. This is visual information. I'm going to send it to the primary visual cortex. Okay. So here's a great diagram of that, figure 6.13. So you have the visual field of the left eye, top left, visual field of the right eye, top right. Okay. What it's saying here is that the right most portion of the visual field of the right eye, you can see how that goes to the medial portion, or the nasal hemiretina, but that would be medial, the nasal hemiretina, and then that transfers over to the left side of the brain. Okay, so it's contralateral. That information goes into the right eye, but it's going to be processed by the left hemisphere. Information on the far left, so now I'm in that pink area, visual field of the left eye. Okay, that information then goes into, again, the hemiretina, crosses over, and is processed by the right hemisphere. Okay. And you see there that you have the eyes, which are the sensory organs. Then they go into the optic nerves. Nerves are bundles of axons. So those are the ganglion cells. Okay. Goes through something called the optic chiasm, which does the crossing over to the other hemispheres. And here's the first time that you get to experience optic nerves, which are part of the peripheral nervous system. Once they hit the brain or spinal cord, which now is the central nervous system, they become tracts. So the optic nerve to the chiasm to tracts. And now those tracts to the lateral geniculate nuclei, and then finally to the primary visual cortex. Okay, retinotopic organization. Information received at adjacent portions of the retina remain adjacent in the striate cortex. Put this a little bit simpler for you, is that there's a representation in the brain, and a lot of the sensory systems are this way. We'll talk about many of them. But so if you have the retina, if you laid out the retina flat, okay, there's a representation in the brain in the primary visual cortex that looks very similar to that. Okay. So that the fovea, which is responsible for high acuity color vision, is going to be represented in the brain by far more brain tissue, far more neurons, okay, than it would be at 30 or 40 degrees away from it, because the brain is trying to devote much more resources, okay, to that information that's coming in from the fovea, and it's understandably why, because it's the most high acuity and color. Okay. About 25% of the primary visual cortex is dedicated to input from the fovea. There you go. Okay. A little bit on seeing edges, which is called contrast enhancement. There's something called these mock bands. They're non-existent. It's something that the brain is actually doing for you. It makes edges easier to see. So let's look at this. Okay. So you see that the blue, it goes from a medium blue all the way out to a very, very light faded blue from left to right. When you stare at that, it looks almost as though there's a white like cloud between each one of the segments of blue. But if you were to put a piece of paper over that, that's not true. Okay? What the brain is doing is it's trying to exaggerate for you the difference between those colors in blue. And so it puts that mock band in there so that you can see the difference. Now, this is a very simplistic idea, but you think about when you look up from the page and look around the room, look at all the edges you have. There's thousands of edges all around in your environment. And that's very important that you're able to identify, oh, this is closer, that's not as close, that's far, far, far away, that's next to it. This is, my computer is sitting just a little bit in front of my speaker system. All those are edges that your brain is interpreting for you. So the idea here is that, yes, you do see the difference between those colors, but the brain, the cortical regions of the brain, not the eye, not the retina, but the cortical regions of the eye are exaggerating that for you. And they believe that it is done through lateral inhibition. Okay, so you see that in this that the edge of those colors are actually right there between D and E. Okay, that lateral network. And so what is there, the physical intensity of the light is the yellow, but the way the cortical regions of the brain exaggerate it, the response of the receptors to the light is represented in the very bottom in there in that dark orange. Okay, and so what they say is that where that edge actually is, that neuron is going to fire very abruptly. Okay, now we know it's all or none, or for those firings, but it's going to fire with intensity there, and so that when that one fires intently, the neurons around it are somewhat subdued. Okay, and it's that lateral inhibition, or the subduing of the neurons next to that neuron that picked up that edge point, is what's causing that mock band for us. Okay. Okay, and now we're going to talk about color. Okay, the component theory, or the trichromatic theory, proposed by Young and Helmholtz. They believe that there were 3 types of receptors, each with a different spectral sensitivity. Okay. And these, I always thought that they were let's see. Okay. So the component theory, the trichromatic theory, 3 types of receptors, and each one of these has a different spectral sensitivity. And the color of a particular stimulus is presumed to be encoded by the ratio of activity in the three kinds of receptors. So this was a theory. It was very early on, okay? And then Herring came around with his opponent process theory, and to understand the color vision a little bit better. He believes that there's two different classes of cells encoding color, and another class encoding brightness. Okay, and the two that he's speaking of are considered a red-green cell, so cells that are specific for red and green, and then cells that are specific for blue and yellow. The idea here is that it's an activation and disactivation, I guess. So if you have the cell activated, it's red. If not activated, it's green. Yeah, and therefore the same with the blue and the yellow. Okay. The interesting thing about this is that this theory accounts for color after images. So you focus on something that's red for a while on a page. Just take a white page and a big red box. Okay, and you focus on that red box for about 30 seconds. As soon as you look away, you'll have a green image that will show in your visual field. And then the same for the blue, yellow. So it does appear that there's an opponent process where if activated red, not activated green, and same blue, yellow. Also, the theory works for this idea of a reddish green or a bluish yellow that don't seem to be, don't seem to exist. Okay, so both theories are correct. Coding of color by cones seems to operate on a purely component basis, and then opponent processing of color is seen at all subsequent levels. Okay, so there's all this on-off happening, but there are also cones that are specific to individual colors. Okay, and then there's another concept here called color constancy. Okay. Color perception is not altered by varying reflected wavelengths. It says color constancy is the tendency for an object to stay the same color despite major changes in the wavelengths of light that it reflects. So let me try to give you an example, and your author gives you several, but you have an apple, and the apple is sitting on your desk, and in the morning you have the sun coming through the windows, and you have a very vivid red apple. Throughout the day, maybe into evening, you still have the blinds open, but there's not nearly as much light. It's kind of a shadow, dark in your room, but you can still tell that is a red apple. Okay? It gets darker and darker. You walk in the room, and you can see it's a very dim environment, but you can still see the red apple. What they believe is that because the wavelengths that your retina is perceiving, right, that's entering into the retina and then being perceived by that as well as the primary visual cortex, is vastly different from morning, afternoon, night. So why are we still seeing it as red? Okay? And what they believe is that it has more to do with the context. Okay? So the primary visual cortex is taking into consideration the context around that apple. Everything's really bright. Red apple, it's red. Okay? And then when you see it again, it's like, okay, well, everything is not as vivid as it was before, so I'm still going to interpret it as red. Okay? But again, and then there's this Retnex theory. We don't go into detail on that. But again, these are theories. They're still under investigation. They're still looking at, but we know that it does work. Color does stay constant, even though the wavelengths change dramatically. So they're just believing that it has more to do with, again, like I mentioned before, what you actually see. What the physical energy in your environment is not necessarily what you perceive. good example of that. Okay, let's look at the flow of the physical energy to the primary visual cortex. All right, so let's do this again. Physical energy in the environment, photons, wavelengths between 500 and 900 nanometers, that's the physical energy. Enters in through the eye. We have the different parts. We have the cornea, we have the pupil, and then it goes back to the retina. Okay, it passes through all those cells. It is transduced by the rods and cones. If those rods and cones are activated, then it's sent back through those cells, bipolar cells, amacrine cells, ganglion cells. Okay, those ganglion cell axons are the feed through the blind spot, and those cells are the optic nerves. The optic nerve then goes to the optic chiasm and switches over to the other side as long as it's being reflected on the nasal hemiretina. Okay, it's sent to the other side. Then we know that it goes to the thalamic relay, which now you have on your slide, and specifically the lateral geniculate nucleus. And from there, then the primary visual cortex. From the primary visual cortex to the secondary visual cortex. From the secondary to association cortices, okay, which we'll see also in a lot of the other sensory systems too, is that there is a primary area, many of them have secondary areas, and all of them have association areas. As the visual information flows through the hierarchy, okay, the processing becomes larger and the processing becomes more specific. And here's a diagram of that for you. So from the lateral geniculate nucleus again to the primary visual, or what they list as the striate cortex, and then from primary to pre-striate, which is the secondary visual cortex, and then from there to posterior parietal cortex, which is one association area, or inferiotemporal cortex, which is another association area. And they have different purposes. So let's look at those different purposes for the secondary. Okay. And I guess, how did we know that there are these secondary, there's these association areas? And one way that we discovered them are through individuals who had these scotomas. And a scotoma is an area of blindness in contralateral visual field due to damage to primary visual cortex. These are discovered through perimetry. Perimetry are tests where they fixate the head. They put the individual into like a chin strap so they can't move their head back and forth. They can only look straight forward, and then they have like little lights that come on, and they have to detect where these lights are. And so they can identify that everywhere where there was a light that came on in the uppermost portion of the right visual field was ignored. Okay, well, there might be a scotoma there in that area then. Patients may be unaware of scotomas also because of, remember, that completion. So their eyes are always moving. They're binocular cues. They're using both eyes. Okay, so it's possible that they're not even aware of the scotoma until they become fixated and go through that procedure, which is the perimetry test. Here's an example here where Lashley actually had a scotoma, and he would not see the head of the individual here. And this happened due to a migraine. When migraines came on, then there was a scotoma in the visual field, and it just happened in his visual field, just happened to be right in the middle. Okay, so whatever was right in the middle of his visual field, he could not see. There's also another thing called blind sight, response to visual stimuli outside conscious awareness. So this is the ability of such patients to respond to visual stimuli in their scotomas, even though they have no conscious awareness of the stimuli. For example, a patient with blind sight might reach out and grab a moving object in her or his scotoma, all the while claiming not to see it. So it's literally a blind sight. Okay. So they didn't go through that test. They didn't know. They asked, okay, can you pick up that object that's sitting right out in front of you on the left-hand side? And they reach out and they grab it. Okay. But they tell you, well, I didn't even know it was there because they couldn't see it. Okay. Again, possible explanations. The second one there, direct connections between subcortical structures and secondary visual cortex. So what they're saying here is that it might be because if there was damage to the primary visual cortex, the secondary visual cortex, the association areas, are picking up some of that loss. Okay? And so they're able to reach out, they're able to grab it, even though they're not fully aware of it. Okay? Because other systems, subcortically, deeper in the brain, are still functional. Neurons in each area respond to different visual cues in these association cortices. Lesions of each area result in specific deficits, and they're retinotopically organized, just like we said the primary visual cortex was. And this picture right here shows you how they've been able to color some of these. So you have like V4, V2, V3, and these are all specific for different things. Okay, it might be for more specific for color, maybe for sizes, for shapes. Okay, but they all have their own, it seems like their own function. Okay, let's talk more about the primary visual cortex and then how that primary visual cortex sends the information to the secondary or the prestriate cortex, and then from there it can either go to the dorsal stream or the ventral stream. Right? So the dorsal stream is the pathway from the primary visual cortex to the secondary to the posterior parietal cortex. Posterior, still in the back of the brain, right? But now it's the parietal cortex. So it's moving out of the occipital lobe, moving into the parietal lobe. So the very back portion of the parietal lobe. This seems to be the where pathway, specific for location and movement. Okay? Or pathway for the control of behavior, like reaching out and grabbing something. The ventral stream is pathway from the primary visual cortex to the secondary to the inferotemporal cortex. Okay? Now think about that. Infero is inferior, that's bottom. So the bottom of the temporal cortex. So now we're talking temporal lobe. And this seems to be the what pathway, specific for color and shape. And the pathway for the conscious perception of objects. Remember how that individual, they can reach out, they have the scotoma, they can reach out and they can grab the object, but they don't know it exists. So through the dorsal stream, they're still functional. There's nothing wrong with the connection between the primary visual cortex and the posterior parietal cortex. But maybe there is a dysfunction between the primary visual cortex and the ventral stream, which is the inferotemporal cortex, because they're not consciously aware of the object. And so here's a nice diagram of that, showing both of the dorsal and ventral streams. And again, it even says, has a prediction there, damage to the dorsal stream disrupts visual spatial perception. Damage to the ventral stream disrupts visual pattern recognition. And then at the bottom, damage to dorsal stream disrupts visually guided behavior, the ability to reach your arm out and grab something. Damage to the ventral stream disrupts the conscious visual perception of that guided behavior. So the exact example that I just gave you. Okay. Then we're actually going to end this chapter with two neurophysiological disorders. One of those is prosopagnosia. Prosopagnosia is the inability to to distinguish among faces. And these are crazy. You can just trying to understand this and how somebody has this kind of a deficit, how they get around in their world. It's pretty amazing. Most prosopagnosics recognition deficits are not limited to faces. So they can also have the inability to distinguish, chairs or homes or something else. Prosopagnosia is associated with damage to the ventral stream between the occipital and temporal lobes. Okay, so now you know ventral stream. Oh yeah, inferotemporal cortex is involved there. Prosopagnosics may be able to recognize faces in the absence of conscious awareness, and that follows along with the same idea with the ventral stream deficit. And then prosopagnosics have different skin conductance responses to familiar faces. So they will tell you, yeah, I can see the face. There's no problem with that. I just can't, I can't perceive the face as anybody who I've ever seen before. Okay, I know it's hard to make sense of that, but yeah, I can detail the whole person. I know what color their eyes are, their nose, their lips, that they have lipstick on, whatever. But I've never seen that person before, even though this person, believe it or not, could even be themselves. With somebody with prosopagnosia, it could even be themselves. Okay, but then when they do this skin conductance, and we talked about this when we were looking at those non-invasive physiological methods, where you put the unit on the little finger and it picks up sweat gland activity, basically. Okay, so if you showed them faces of people that they were familiar with, this skin conductance response would go off. So obviously, they do their bodily system somewhere else is saying, yeah, you know this person, but it's just because of the disorder. It's because of the disruption in this specific area, this ventral stream, that is not allowing them to do it. Okay, so it's very, very complex. This is something, again, that a clinical neuropsychologist would work with. This could have been due to a stroke issue or who knows what. And like we mentioned before, the neurosurgeons are out of the way. The neurologists are out of the way. We're not dealing with a chemical issue here, so we're not dealing with neurochemistry. We're really dealing with how do we help these individuals go about in their daily life. Okay, so this is a prime example of something that would be studied by a clinical neuropsychologist.